Chapter 4 is genes and cellular function. Last week we went over macromolecules and we talked about proteins and why they are so important in our bodies. This is just a little review of what proteins do. So proteins can be digestive enzymes and help facilitate chemical reactions. They can become antibodies. They can support muscle movement. They can be useful in expressing DNA. They can provide muscles or bones or tissues for the body. They can help our hormones, and they can also move things around in our body. So proteins play tons of very significant roles inside of our bodies. So DNA, deoxyribonucleic acid. It's made up of nucleic acid. That's why it's called deoxyribonucleic acid. We talked about that last week. But DNA is the genetic makeup of our body. All living things have DNA. So it is a long thread-like molecule with uniform diameter but different lengths. So width or around, it's the same size, can be all different kinds of lengths. Humans have 46 DNA molecules or chromosomes in the nucleus of the majority of our cells. Okay, these 46 chromosomes come in 23 pairs. So a pair of 2, 23 of them. Typical human DNA is about 2 inches long. And again, they are made of nucleic acids, which make nucleotides, and it's polymers of nucleotides. That's the building blocks of DNA. So what does the nucleotide structure of DNA look like? So it's made up of nucleotides that consist of a sugar and a phosphate, as well as a nitrogenous base. So the sugar in DNA is deoxyribose, which is why it's called deoxyribonucleic acid. It says it right in the name. It's deoxyribose. And then there's a phosphate group attached to that sugar. Attached to the other side of the sugar is a base. The base can be one of 4 bases. It can either be adenine, thymine, cytosine, or guanine. And we refer to them as A, T, C, and G. Okay, so if you look at these nucleotides, there's a sugar with a phosphate attached to one side and a base attached to the other side. That's the basic structure of the nucleotide for DNA. So going into a little bit more detail about these nitrogenous bases. So they're called nitrogenous bases because they have nitrogen in them. It's primarily nitrogen, or their base is nitrogen. So you can be either a double ring or a single ringed nitrogenous base. If it's a double ring, it's a purine. Okay, adenine and guanine are double ringed, A and G. Okay, and if you look at the picture, you can see that the purine has 2. Pyrimidines are single ringed bases. These ones are cytosine and thymine, the other 2 for DNA, and then uracil, which is specific to RNA only. It's not in DNA. So just hold on to that, and we'll get to that in a second. But the double ring is A and G, and the single ring is C and T. So if you've ever seen any pictures of DNA, it looks like a spiral staircase. Okay, that structure is called a double helix. So looking at the actual makeup of that double helix, going back to the nucleotides, we have a bunch of nucleotides that connect together to make the backbone. And in the middle, the bases will bond with their matching pair. So the sides of the DNA are the sugar phosphate backbone. In the middle, the little like ladder crosses in the middle, those are the base pairs. So the base pairs have to pair up with a complementary pair. So when you look at DNA, there are 2 backbones, and then the base pairs meet in the middle. Those base pairs meet in a very specific way. So they can only pair with their actual complementary pair. So when things pair together, a purine is always going to pair with a pyrimidine. Okay, a single ring is going to pair with a double ring, and they're going to pair in the same pattern always, where A pairs with T and C pairs with G. So adenine to thymine, cytosine to guanine. It will never pair any other way. A will never pair to G. A will never pair to C. It's always going to be A, T, C, and G. And you can remember this by apples in the tree, cars in the garage. So A to T, C to G. When A and T bond, they have 2 hydrogen bonds, and C and T make 3. Okay, it's just because their structure, they have different bonds. But a DNA molecule is 2 backbones of the sugar phosphate and the base pairs connecting in the middle, in the same pairing pattern, basically, of A to T and C to G. So the base pairs are always going to pair A to T and C to G because of the law of complementary base pairing. So purines will always pair with pyrimidines, and A will always pair T, C will always pair G. That's just how it is. And if you look at this picture, you can see that the A and the T are kind of rounded, and the C and the G are kind of triangular. They won't pair any other way except for A to T and C to G because that's how it's made. Okay, so when you're making a sequence of DNA or writing a code for DNA, make sure you know that A and T go together and C and G go together always. So a gene is a segment or a chunk of DNA that codes for the synthesis of a very specific protein. Okay, and then a genome is all the genes inside of one person. We have about 20,000 genes in our body, and only 2% of them are actually DNA. The rest play other roles in the body that aren't for coding, okay? So genes are just a chunk of DNA that tell a ribosome what protein to make. So another part of DNA is a chromatin. So a chromatin is a fine filament-like DNA that it has proteins embedded in with it. So there are 46 chromosomes in most cells. They're about 6 feet long. And in non-dividing cells, so cells that do not undergo mitosis or cell division, they're really, really small, and you can't really see them with a light microscope. They move around a lot. And these things, the chromatins, when you look at them or you see a picture of them, it looks like beads on a string. So if you look at this picture, you can see that there are these orange little ball things, and they're wrapped with purple. Those are the beads on a string that make the chromatin. So the orange ball wrapped in the purple is the actual chromatin. So what is this orange ball wrapped in this purple DNA? So the chromatin is a group of proteins, okay, 8 proteins specifically, that are wrapped up in a DNA strand. Okay, so these proteins called histones cluster together in 8, and then DNA wraps around it 1.5 times. Okay, when it wraps around it 1.5 times, it'll move on to the next one and wrap around that one 1.5 times. Then it'll move to the next one and wrap around that. Okay, so it'll keep doing that until it has this chunk that it needs, and that's the chromatin. Okay, in between each of these histones, there's a little piece of the DNA that is not actually wrapped around that cluster of proteins, and that is called the linker DNA because it links them together. Okay, one bundle of those histones with DNA wrapped around it is called a nucleosome. So we have histones and DNA. When you get a cluster of histones, 8, and DNA wraps around it, it forms a nucleosome. When it wraps around the next one, and the next one, and the next one, it forms a chromatin. It's a group of nucleosomes. Okay, and in between each nucleosome is a little strand of DNA that's not wrapped around anything, and that's the linker DNA because it links them all together to make that chromatin. Chromosomes. So we talked about chromatin, now we're going to talk about chromosomes. So each chromosome is packed into the nucleus in a very specific area. Okay, it has a chromosome territory, it stays in its area. This area can be permeated with channels, allowing for chemicals to come in and out to access or turn on the genes. But it's a highly protected area because we don't want the chromosomes just leaking out to the cell and going everywhere. They need to stay together, they need to stay in their area. In non-dividing cells, the chromatin move all over the place, and genes can be turned on and off depending on the needs of the body. So just because you have a gene doesn't mean you're going to have that gene always functioning or always turned on. Some genes always stay on, like the genes that tell you that you're a female or a male. Of course, that's always going to stay on because that's just who you are. But there are some genes, like the gene to produce breast milk. Those are going to be ones that turn off and only turn on when the body needs them to turn on. So RNA, ribonucleic acid, is very similar to DNA, but it has a pretty different job. So back when we talked about base pairs, the purines and the pyrimidines, I mentioned uracil, and I said it was only for RNA. So RNA is similar to DNA in the fact that it has a sugar, but it's not deoxyribose, it's just regular ribose. And instead of having thymine, it has uracil. So there's no T in RNA, it's just U. Okay, RNA is a lot smaller than DNA, and it's also typically a single strain. Okay, it can be double, it can pair with itself, but usually it's just one. Okay, so the main function of RNA is to translate the genetic code to make proteins, and it functions mostly in the cytoplasm or the jelly center part, not the nucleus, the jelly middle part of the cell. Okay, and in protein synthesis, there are 3 RNAs that are important. We have mRNA, rRNA, and tRNA, messenger, ribosomal, and transfer RNA. Those are the three that are important for protein synthesis. Okay, going back to genes. I told you they were a chunk of DNA that coded for a protein. A little bit more in detail now, since we have a little bit more information. They are actually a chunk of DNA that codes for an RNA molecule to be made, and then that RNA molecule will tell the ribosomes what protein to make. Okay? So an amino acid sequence of a protein is determined by the DNA sequence. So DNA writes the code for which proteins are to be made. And again, in humans, we have 46 chromosomes, 23 pairs. Okay, and the genome is all of our chromosomes, like all the chromosomes in DNA in one body. And genomics is the study of the whole genome, like the study of chromosomes and human genes and how they interact to affect organisms. So the genetic code, just like computers have codes to run, our body has a genetic code to run and create things. Okay, so the genetic code is made up of DNA, and it's nucleotides that code for amino acid sequences of proteins. So remember, proteins are made up of amino acids, and the DNA tells which amino acids come together to make that special protein. Your body can make millions of different proteins just from 20 amino acids. We only have 20 amino acids, and we can make millions of proteins with different combinations of these amino acids. Okay, and these are made from just 4 nucleotides, the A, T, C, and G. Those are the only 4 nucleotides that we can get from DNA, and they can code to make millions of different proteins. Okay, a minimum code to make a 20 amino acid chain is 3 nucleotides. Okay, so 3 nucleotides will make 1 amino acid, and then we can make a 20 amino acid chain. So here's those 3 nucleotides that make the code for an amino acid. So the 3 nucleotides are called a codon, okay? And each one of those nucleotides within that codon is a base triplet. They're called base triplets because it takes 3 of them. It's a triplet. So if you look here, we have this RNA strain, okay? And this read the DNA, and it pulled the code from it, and now it gave us this code over here on the side. So CUU is a base triplets. There's 3, and those make 1 codon. CGG makes the next codon. AGC makes the next codon. Okay? So the way this works is the RNA goes to the DNA, and it says, okay, what do you have? It reads the DNA sequence, and it puts its complementary base pair to it. So if we have DNA that's TAC, okay? Well, we know that the complementary pair for T is A. We know the complementary pair for A is U, because remember, RNA is uracil, not thymine. And then we know the complementary pair for C is G. Okay, so it would make that code, and that would be what the RNA has. That would be 1 codon. Okay, so the RNA is going to read the DNA, get its code, and make codons. And those codons code for the protein. So which DNA does the RNA know to read? The DNA has specific codes in it that tell the RNA to start and stop, and what to read in between to make the proteins. So there are 64 possible codons available, and these represent 20 different amino acids. 61 of those make proteins, and 3 are stop. 3 tell it to stop, we're done, we don't need to make any more. So the start codon is AUG. So anytime there is the code AUG, that means start making a protein. And then the stop codons are UAG, UGA, or UAA. This means stop making the protein. So the RNA will transcribe the code from the AUG all the way through the stop, and everything in between is what makes that protein. Once it gets to the stop, it's done. So it's only going to read from the start to the stop. Nothing after and nothing before. So in our bodies, all of our cells, except for our sex cells, have the same genes. They're identical. Okay, there are different genes that are activated in different cells depending on the function, and cells only use about 1 to 2/3 of their actual genes. The rest just lay there and do like regulatory functions, or they lay dormant and don't do anything until they're needed. Okay, so all this information about DNA and RNA, let's put it together and talk about how it actually makes proteins, because it's not important unless it can make the protein. So protein synthesis, or the making of proteins, is basically DNA translating to RNA, and then RNA translating that to a ribosome to make a protein. Okay, so this occurs in 2 steps. There is transcription and translation. Transcription comes first. This is when RNA takes the code from DNA. Okay, this happens inside the nucleus. And then translation happens after that. And translation is when RNA tells the ribosome the code to make the protein. And this happens in the cytoplasm. Okay, so in a quick rundown, we have DNA, which is then transcribed into RNA, which is then cut and spliced to make the right code that it needs, leaves the nucleus. Okay, then it comes out into the cytoplasm, finds a ribosome. The ribosome then reads the mRNA, and that tells what protein to make. Okay, so DNA talks to RNA, and RNA talks to the ribosomes. So transcription is the first step in the protein synthesis, and this is when the RNA enters the nucleus and copies the genetic instructions from the DNA. Okay, certain DNA bases function as a signal start. So mRNA is going to go in and look for the base that says start, and it's going to start copying from there. And if you think about what the word transcription means, or to transcribe, it means to make a copy of something. So the RNA is going in, reading that DNA, and making a copy. The RNA that does this in transcription is RNA polymerase. Okay, so the RNA polymerase enters the nucleus of the cell. It binds to the DNA and opens it up so it can get in and read one side of it. Okay, it's going to read the side and make the complementary strand. Okay, if it sees a C, it's going to put a G. If it sees a G, it's going to put a C. If it sees a T, it's going to put an A. And if it sees an A, it's going to put a U, because remember, there's no T in RNA. Okay, so RNA polymerase goes in, reads the DNA, and makes its complementary strand for that code. So after the RNA has made its strand, it is now considered pre-mRNA. It's not ready yet, so it's not mature, but it's it's almost there. Okay, once it has this code, it needs to be kind of edited a little bit. So it's immature, and then enzymes in that nucleus will come to it and cut things out, remove things, or trash things. Okay, this is called splicing. Okay, it's going to come in and pull out chunks that are not needed. Okay, so there can be exons or introns when we're talking about splicing. So exons are segments of the mRNA that are going to be exported from the nucleus and then translated into protein. So these are going to be used to make proteins, but not in this chunk of the mRNA. Introns are trash. So the introns are pulled out and then they're just thrown away. We don't need them. They're not useful. So after the splicing happens in the nucleus, the mRNA can be pieced together in a specific order to code for a specific protein. So many different variations can happen in this alternative splicing method. And if you look at the picture, it kind of illustrates for you. So the pre-RNA was red, yellow, green, blue. Well, in this first, they took out the green and they made red, yellow, blue. Well, that can code for this type of protein. Or on the other one, they took out the yellow and they made red, green, blue. Well, that can code for a different protein. Okay, so splicing together things in a different way, different chunks, can make different proteins. And here's a quick little rundown again. So we have a DNA molecule. It is copied by RNA. It makes pre-RNA. Remember, it's not ready. It needs to be spliced. So it's spliced. It's spliced and made into whatever little sequence that it needs to make the proteins. So in this case, it was spliced and made into ACD, BDE, and AEF. Okay, each one of these sequences codes for a different protein. Okay, so ACD codes for protein 1, BDE for protein 2, and AEF for protein 3. So all from one strain of mRNA, we got 3 different spliced codes to make 3 different proteins. So now we have this mature mRNA. It's been spliced. It's been pieced together. It's ready to go. So now we're going to do translation. So after the mRNA is made, it's time to translate. So the mRNA is going to leave the nucleus after it copied the code. It's all ready. It's going to leave the nucleus. It's going to go out to the cytoplasm, and it's going to find a ribosome. Okay, it's going to bind to that ribosome, and then it's going to translate that DNA language that it just copied into protein language for the ribosome to create the protein. So the ribosome and the DNA do not speak the same language. The RNA has to be the translator. Okay, the DNA cannot go to the ribosome and say, make this. It has no idea what it's talking about. So DNA language is converted into protein language by the RNA. So, and there are three major things that play a role in translation. First, we have the mRNA, because that's what copied the code from the DNA. So that's already got the code. It's carried it out of the nucleus into the cytoplasm, and it's looking for its specific ribosome. Okay, then we have the transfer RNA. The transfer RNA is told by the ribosome, based on the code from the RNA, which amino acid to go get. It goes and gets the amino acid, and it brings it back to the ribosome. Okay, it can go back 30 times. 20 different tRNAs can go get these amino acids and keep bringing them back. But it's transfer RNA, so its job is to transfer amino acids from the cytoplasm into the ribosome to be built into proteins. And then, of course, the ribosome plays a huge role in this part, because that's what actually builds and puts the proteins together. So the messenger RNA, after it comes out of the nucleus, is going to go into the cytoplasm, and it's going to find the specific ribosome that it needs to bond with or attach to. Okay, there's going to be a flag that says, hey, this ribosome is what you need. They only go to specific ones. Okay, it's going to act as a binding site for a small ribosome, the subunit or part of a ribosome, and then a bigger chunk of a ribosome is going to come attached to it. Okay, they're going to basically sandwich that RNA in it like a ribbon, and it's going to pull it through and read it. It's going to keep pulling it through and reading it until it finds a start codon, or AUG. As soon as it sees that AUG, it knows it's time to start building. Once it reaches AUG, it's going to find a tRNA and say, hey, go get me this amino acid. Okay? Once the tRNA leaves, we're no longer dealing with the mRNA. We're just dealing with the tRNA, picking up the amino acids. So once that start codon happens, tRNA is activated to go get these amino acids. The tRNA is going to go out to the cytoplasm and look for the amino acids that it's being told to gather so they can build a protein. Okay, tRNA is a funny shaped coil with an empty end. Basically, one end has a binding site and the other end has an anticodon. So an anticodon is a sequence that tells it which amino acid it needs to go get. So one amino acid can bind to the end based on that code. It can only pick up the amino acid that it's looking for. It will not pick up the wrong one on accident, because only one kind can bind to that end. Okay, and when these tRNAs go out and find their amino acids and bind with them to bring them back to the ribosome, it does require energy, and it takes one molecule of ATP for it to go get it and bring it back. Okay, remember, these amino acids are in the cytocell, or the liquid and fluid in the cytoplasm of the cell. So the tRNA is told by the RNA, go get this amino acid. It leaves and goes and gets it, and it brings it back to the ribosome so it can connect it to another one. So after all these tRNAs go out and they get all these amino acids, and they have this protein chain built, they're eventually going to hit a stop codon. That mRNA is going to say stop, and the ribosome is going to say, okay, we're done. No more tRNAs going out. We have everything we need. Okay, so once that chain is built, the proteins are either sent to the endoplasmic reticulum to be folded and made into their structure so they can be functional, or they go to be exported out of the cell. So it just depends on the type of protein, what's going to happen. Some proteins can be lysosomes, or the little destroyers in the cell, the ones that get rid of the waste or eat parts of the cell that are no longer functioning. Or they can even be made to secrete and come out of the cell as secretions. So newly made protein goes into the rough endoplasmic reticulum, where it's modified, bent, and packaged, basically. So transcription is just one main step. The RNA goes in, reads the DNA, copies it, it gets spliced. But translation has three. So the first step in translation is initiation, which is when it starts. Okay, and it's initiated by that start codon. Okay, So you have the RNA comes out of the nucleus. It finds a small chunk of the ribosome, bonds with it. Okay, it binds to it. Then the tRNA matches with the mRNA and reads it until it sees a start codon. Okay, once it finds a start codon, the large RNA or the large ribosomal unit will come and join on, making this fully functional ribosome. So the ribosome comes in two subunits, large and small. The mRNA and the tRNA are going to come to that ribosome. They're all going to be together. So mRNA, the large unit, the small unit, and the tRNA. That's initiation. So nothing has really been read except for the start, and it's connected together. So the actual process of the tRNA going out and getting the amino acids and bringing them back to the ribosome, that's called elongation. Okay? So in elongation, we're adding more amino acids onto this peptide chain. So we're making it longer. So the tRNA will go out and get this amino acid. Then another tRNA will go out and get this one. And then one will go out and get this one. And they'll keep getting them as long as that RNA tells them to go. Once the stop codon is reached, it'll stop. Okay? This will continuously repeat until it gets to the stop. And then when you get to the stop, you should have the chain that you need to fold your protein. The last step is termination. This is the end. When it gets to the stop codon, it stops, it terminates. We already have our protein chain. We're done. So what happens then is the protein breaks away from the ribosome, and then the ribosome breaks back apart into those two units that it was originally in before it came together on the RNA like a ribbon. Okay, so protein goes away, it breaks off, and the ribosome breaks off into its two units like it was already in before. And to just get an idea of how fast this happens, translation is really, really fast. We have 300,000 RNA molecules undergoing translation at the same time in our body inside one cell. So a cell can produce 100,000 protein molecules every second. So this is not a slow process. Like, it can happen really fast, and there's millions and millions of things going on at one time. Okay, so translations, super quick, happens all the time. We're always making new protein. So here we have like a easy rundown of actually how DNA writes for the peptide sequence. So how does the DNA code tell what protein to build? So the DNA's double helix is then copied into base triplets to form the RNA strand. Okay, that RNA strand is then spliced, put together, and it comes out to make this code, which is initiated by the start codon, and it's ended by the stop codon. Okay, once those are read by the ribosome, it signals the tRNA to run out and pick up a certain part of a protein or a certain amino acid based on the anticodon on the tRNA. It brings it back to the ribosome holds that one, and then the next tRNA goes out and gets another one, and it comes back, and then it attaches both of those amino acids together. This continues all the way down that code until you reach the stop codon. Once that stop codon is reached, no more amino acids are picked up by the tRNA, and that's the complete protein. After that, it breaks off of the ribosome, and then the ribosome disassembles back in its two separate units. So after the amino acids have been put together in this peptide chain, they're not ready for use. If you remember, proteins are really intricately folded and bent, and in order for a protein to work properly, it has to be in that very intricate structure. So once the protein chain is made, it has to be folded. So after it's made, it leaves the ribosome, and it goes into the rough ER, which normally it's already in the rough ER because the ribosomes are on the rough ER most of the time. They're built there. So it goes to the rough endoplasmic reticulum, and then it will fold and bend it in whatever way it needs to be folded and bent. Okay. Usually, things called chaperone proteins will guide this protein on how it should be bent. The chaperone protein will tell it, you're supposed to be this kind of protein, and this is the directions you bend this way, then this way, then this way, and so forth, to get it to bend into its proper form. So the proteins that are created in the ribosomes that are in the cytosol or the cytoplasm are usually going to be in the cytosol or cytoplasm. Okay, they're usually used there. They're made there and they stay there. So proteins that are made in the ER, or the endoplasmic reticulum, are usually sent to the Golgi after they're folded, so the Golgi can package them up and inspect them and make sure they're proper. And usually those ones get shipped out of the cell for use elsewhere. But they're shipped from the rough ER to the Golgi, and the Golgi finalizes them and packages them, inspects them to make sure they're correct. So proteins that are assembled in the ER, or the rough ER, are threaded through a pore in the ER's membrane into the cistern, which is the big area inside that holds the proteins. Okay, it holds them and it takes them where they need to go. So enzymes inside of the cistern will modify the protein by either removing the amino acids that are not needed, folding them more, stabilizing them, adding carbohydrates, adding lipids, whatever needs to be added or done to this protein is usually completed most of the time, at least 90% of it, completed in the cistern of the ER, and enzymes play a really big role in finishing that process. And this is called post-translational modification because it happens after the translation process. So when the ER, or the endoplasmic reticulum, is finished with the protein, it pinches off a bubble-like vesicle known as a transport vesicle, and it takes it from the rough ER to the Golgi. Okay, so it's a little bubble that breaks off of the rough ER and goes to the Golgi with the proteins inside of it. Okay, and then to simplify this process, ribosomes make the proteins. The rough ER folds and modifies them and gets them in their really fancy structure. The Golgi sorts them, packages them, and inspects them, gets them ready to ship out of the cell. And then the Golgi vesicle will transport them out of the cell or to other parts of the cell where they need to be at. So once the protein gets to the Golgi, it is either fused or separated. It can be modified, more complex, so it can be way more intricately modified in the Golgi. It can add carbs, it can add other glycoproteins to it. Sometimes it becomes lysosomes, but when the Golgi is done with this protein, it's going to do the similar thing by breaking it off into a little transport vesicle, and then it's going to move it to where it needs to be. It can either go out of the cell or a different place in the cell, but the Golgi finishes up the protein. When the Golgi is done, the protein's ready to go, and it moves it in a transport vesicle just like the ER does. And the last thing we're going to talk about is that genes can be turned on and off. So just because you have a gene in your body doesn't mean you're always going to display that gene. Some genes are going to be on all the time. Some genes are going to be off all the time. It just depends on what the gene is. Some genes can be turned on and off when our bodies need them. So certain genes, such as mammary glands, those only turn on during pregnancy or after birth. They're not on all the time. Okay, so all of our genes are able to turn on and turn off, and 90% of the time, if not more, they are controlled by hormones in our body, which are chemical messengers. And we'll talk a lot about how these hormones control our genes when we get to the endocrine system.